Hi all, welcome back to Microbiology Lectures, and we're going to continue talking about cytokines or the adaptive immune response. So last time we talked about different ways at which cytokines can kind of be organized, whether it is virus-based or interleukin or tumor necrosis factor or just different ways that we kind of label them. And this time we're going to talk about what happens to those signals. So remember, cytokines are little proteins that are sent out, and either they can be sent out via just sent out via blood or fluids in the body. And we often think of them kind of like a voice. Okay, it's like it's a signal telling other things about some kind of infection or damage, or there's a virus here, there's a bacteria here, these different things. There's signals, okay, that are sent out. Now, they're sent out in kind of two ways. They can be sent through the fluid, or they can be held on to in cell-to-cell communication. Okay, so when we look at this, cytokines are often grouped into what kind of response is going to be given. So we have, for example, like tumor necrosis factor are what we call pro-inflammatory cytokines. So when a tumor necrosis factor is detected, the body will send more fluid, more blood cells, and both red and white and platelets to that region. So inflammation occurs and more fluid is sent to that region. You have also anti-inflammatory cytokines, which do the opposite. It constricts and it doesn't allow fluid to go to that region. Okay? And so many of these work together, or they can act on together to get a response. So you might have interleukin signals being sent, tumor necrosis factors being sent at the same time, and depending on what the purpose or what kind of response is generated, you could have pro-inflammatory cytokines being sent to the same area, and lots of them. Okay? And in fact, this is what we were seeing with COVID-19. For some individuals, you were having multiple cytokines, multiple different types of cytokines, which having the same general response were being sent to an area. So say the lungs, because the virus was getting in the lungs, and you'd have these pro-inflammatory cytokines being sent to the lung tissue. And so you have a massive amount of fluid rushing into the lungs. Okay? And this was causing complications. And it wasn't just the lungs, it was liver and kidney, and there's all kinds of places at which you can have these responses, but we call it a cytokine storm when you have a bunch of cytokines, different types of cytokines, all working basically together, like they're all pro-inflammatory, and so they are all sending fluid to one region, or maybe they're all anti-inflammatory and not allowing blood flow into a certain region. And so you can have a potentially deadly effect from those cytokine storms. And that's what we're seeing with some patients in COVID-19, was that you would get this huge cytokine storm that was rushing in and destroying tissue, organs, etc., in response to the viral infection. Okay? All right, so now you have these cytokines that are being sent out, and we talked about complement a little bit. Now, complement is a series of proteins that have different responses based on the signal that's being collected, based on the cytokine that's being collected. Normally, when we're talking about complement, we talk about C1 through C9, okay, and they're ordered based on when they were discovered. But C1 through C9, and just realize this is totally species dependent. This is a broad overview of how the complement system works. We actually have far more than 9 complement proteins. In fact, some estimates are we have 30 to 50 complement proteins, and many of which can be split into fragments, and often split into fragments because they're not stable. Like C3 splits into A and B because it's not a stable protein. But regardless of that, this is just kind of an overview. If you want more information on how complement and the immune system works from that perspective, you should take an immunology class. And so complement, again, it's proteins. It's part of the adaptive immune system, and this series of proteins have kind of 3 different pathways at which can trigger different types of responses. Okay? And so we're going to talk about the 3 different pathways, and then we're going to talk about what response you could expect from each one of those pathways. Okay, so the first pathway, which we call that the alternative pathway, that's triggered when C3b binds to a foreign cell's surface. Okay, so C3b is this split of C3, and there's an A part and a B part. If C3b binds to a foreign cell surface, okay, it will induce phagocytosis. It induces phagocytes to consume that material, and we'll come back to that. Okay, there's always C3b present in at least some amount, because C3 is not very stable, so it's unstable, so it'll break apart, and you'll always have C3b. Other than that, you can also have some of the other complement proteins triggering the splitting of C3 into an A and B. Okay, and for more information on that, we can talk later. Okay. The lectin pathway is recognizing what we call MBLs, or mannose binding lectins. Okay, these are typically found on microbial cells, and so it's an interaction. So you have the mannose of microbial cells is bound or allows for lectin to bind to it, and when that happens, it triggers a complex. It triggers a response. Okay, and then the one that's probably the most familiar and the one that we often talk about the most is the classic pathway, and this is kind of what I've been hinting at, throughout these different lectures. You have these antigens that activate antibodies, and then when the antigen is present on the outer surface of some pathogen or foreign material, the antibody then attaches to it, and that creates this ability for the cell to be destroyed. And so it's actually the cell membrane is, the holes are punched in the cell membrane, and it's destroyed. Okay, so 3 kind of ways in which the complement system works. You can have the alternative pathway, the lectin pathway, the classic pathway. Now let's look at how those will activate different major outcomes. Okay, so again, this is just an overview of how this works. Opsonization is when C3b binds to the outside cell, so like a bacteria cell, and that will promote phagocytes to attach to the opsin, okay, and then consume the material. Okay. So this is going to, that C3 C3b binding is going to induce more phagocytes, so your neutrophils, your basophils, okay, your eosinophils, to come to the region and consume that material. Inflammatory response, C5a, which will also attract phagocytes to the area. So again, C5 has a relationship with C3 in that it can bind up C3a or be part of the C3a, C5a pathway. Well, that increases the permeability of the blood vessels. So we kind of talked about this in the original lecture. Okay, so when you have a cut or you have a wound, and that wound it needs to have things like thrombocytes go to heal the wound, or platelets to go and stop the platelets, and then the blood vessels need to be permeable and allow for phagocytes to move to the region to consume bacteria or viruses or whatever it might be, some foreign body. Okay, this is where that inflammatory response would come from. The C5a complex would be that inflammatory response. Okay, it also causes those mast cells that are typically associated with tissues to release cytokines. After they consume material, they'll release cytokines, which again triggers more phagocytes to the area. Okay, and then because it's called the classic pathway, you must assume that most of the complement proteins are related to it, and you would be right in thinking that. So C5b, 6, 7, 8, and 9 molecules all assemble in the cell, and it causes the cell membrane to basically holes to be punctured or lyse the cell membrane. Okay, and we call that a MAC attack, or membrane attack complex. So we normally call that MACs, or some people call it MAC attack, and that's to lyse the membrane, and that's normally induced by an antigen-antibody relationship. Okay. The final one that I want to talk about is fevers. So we talked about fevers before, and it's this kind of unique capability. Not that other organisms don't do it. I talked about how fish can induce their own fever by moving into warm water, but humans are really good at this. Birds can do this also, but mammals and birds are really good at trying to increase their body temperature to fight off pathogens, and that's exactly what is going on. So pyrogens are specialized cytokines that induce fevers, so they increase the body's normal temperature. So our normal temperature is about 37 degrees Celsius. Okay, and when you have these pyrogens that are in circulation, it triggers the body to increase its body temperature. Use the metabolism in the body, the energy in the body, to increase its temperature. Okay? And there's lots of reasons for that. Again, if you can get that temperature of the body to be over the microbe's optimal temperature, then that microbe will start to die before the body starts to die. Okay? And we've talked about this before. We talked a little bit about this, but there will be like a threshold even for your proteins. The enzymes in your body will have a certain threshold, an optimum, and over that optimum you'll start to see denaturation. The proteins will start to unravel, and then when they reform, they don't reform correctly. But if you can just induce a fever, even 1 degree over your normal temperature, that might be enough to reduce the growth rates of bacteria. It could actually cause the bacteria to its proteins to denature, and it allows for more time for defenses to occur. So it's not just trying to kill the bacteria. I mean, that's part of it. Like, if the bacteria can't grow in that temperature, then great. But if the bacteria slows its growth in that temperature, then that's maybe enough time for your innate immune system and adaptive immune system to fight off whatever that infection might be. Okay, the other thing that it can do, so when you increase temperature, it can increase rates of enzymes that might be used to break down things like the peptidoglycan layer. So enzymes like lysosomes may work just a little bit better when you increase the temperature just a little bit. So there's lots of ways at which this can occur. Okay, in some cases it can increase the multiplication of lymphocytes, increase the release of leukocytes from the bone marrow. So there's lots of ways in which this induced fever or this increase in temperature can cause the body to start producing more immune cells. It can cause the growth rate of certain things like bacteria to decrease, and overall increase the ability for the body to fight off whatever the foreign material is. Okay, so with that, that's it for kind of the innate immune system and adaptive immune system together. And we're going to talk more about the adaptive immune system next time we come back.